Assalamu alaikum, respected participants and my dear fellows. I would like to start my talk by thanking AZSP for arranging ICAZ-2020 virtual conference. My name is Ammara Riaz and I am going to talk on the topic screening of natural products against human triple negative breast cancer cells. Cancer is a complicated disease marked by the deregulated epigenetic, metabolic, and molecular signaling pathways. This is one of the most fatal diseases in the world, affecting about 14.1 million people annually. The most dangerous types of cancer include lung cancer, liver cancer, colon cancer, and breast cancer. The topic of my research mainly focuses on breast cancer. This is because Pakistan has one of the highest incidence rates of breast cancer in Asia. This disease kills 40,000 Pakistani women annually. We can see that 1 out of every 8 women suffers the risk of developing breast cancer throughout her life. There are 4 other subtypes of breast cancer, but I have specifically focused on triple negative breast cancer. Now you people may ask that what is triple negative breast cancer? Actually, there are 3 important receptors in breast cancer cells, namely estrogen receptor, progesterone receptor, and human epidermal growth factor 2. In case of TNBC, all of these characteristic receptors are absent, making the situation challenging for the researchers as well as clinical practitioners. So the next question is why we are focusing on triple negative breast cancer. This is because it has high mortality rate, poor prognosis, and there are no available drugs for treatment, as well as it is resistant to currently available chemotherapy. Shortly, we can say that TNBC is a highly aggressive subtype of breast cancer with no currently available chemotherapeutic biomarkers. Review of literature has enabled us to formulate the hypothesis that natural products may have the potential to cure human triple-negative breast cancer cells. Keeping in view this hypothesis, we have formulated the objective of our research to screen the plant extracts and natural compounds to identify TNBC growth inhibitors using TNBC, that is MDA-MB231, and non-TNBC, that is MCF7 cell line models. To fulfill the objective of our research, we have formulated a screening strategy to determine the anti-cancer potential of natural products against TNBC. For this purpose, we have screened 2 types of natural product libraries, that is plant extract library and natural compound library via MTT assay. Plant extract library was established by collecting different type of plants, drying them under shade, and making their extracts using soft-slate apparatus and rotary apparatus. Natural compound library was established by collecting different type of natural compounds from customized natural product libraries. The protocol of MTT assay has also been shown on the right side of this slide. This slide shows the screening results of different plant extracts against human TNBC cells. The graph on the left hand side shows that Mangifera indica seed extract is most potent against human TNBC cells, followed by Mangifera indica bark extract, Eucalyptus extract, Aloe vera extract, and Caltropis leaf extract. Here it is to be mentioned that the plant extract, which inhibited the cell viability by more than 40% at a concentration of 100 microgram per ml, were considered as positive hits. The graph on the right hand side of the slide shows the comparative cytotoxic behavior of these plant extracts. IC50 values of these plant extracts has also been shown in this slide. As we have seen that plant extract library, from the screening of plant extract library, we have obtained 5 potent plant hits against human TNBC cells. The next step is the screening of natural compounds against human triple negative breast cancer cells. From our research, we have identified 4 potent plant hits which were found effective against TNBC cells. When these compounds were further screened, we found that 2 natural compounds were selectively effective against human TNBC cells, while the other 2 plants were not selective against human TNBC cells. This slide shows that out of 100 natural compounds screened, only two compounds, ariflexin B and resistomycin, the chemical structures of which have been also shown here, were effective in controlling TNBC cells. IC50 values of these two compounds have also been shown here. These two compounds have been recently reported by me in my research papers. There is also a graphical representation of anti-TNBC activities of radio B and resistomycin. Now let me tell you about the novelty or significance of my study. In this study, 51 plant extracts were screened, out of which 5 plant extracts, that is Mangifera, indica, seed and bark extract, Eucalyptus extract, Aloe vera extract, and Caltropis procera were found effective against cancer cells. This study is one to report two novel compounds, that is ariflexin B and resistomycin, which exhibited anti-TNBC activity. In vivo studies and toxicological profiling are required for further investigations. These are the references I have used for making this PPT. Thank you so much for your attention. Allah Hafiz and stay blessed.